Okay, guys. So today this is going to be part one of blood, um, and this is also the first chapter of the cardiovascular system. Um, so when we look at blood, blood is actually a connective tissue. It's a liquid tissue that consists of plasma, which is a liquid portion, along with formed elements. There are about 5 liters of blood in a typical human body. That makes up about 8% of the total body weight. And blood is continuously moving. It is circulating throughout the blood vessels at all times. Now, if you were to take a blood smear and actually look at it, what you would see is primarily going to be erythrocytes, as you can see here in this image, where the erythrocytes are all of your red blood cells. And that's what all these little red cells are, okay? Occasionally, you might see a big purple stained cell, and we can see 3 of them here. These are 3 different uh, excuse me, 3 different leukocytes, which are white blood cells. Okay, and then finally, you might see tiny little fragments scattered throughout as well, and these tiny little fragments are going to be platelets. But the predominant cell type that we do find in blood is going to be erythrocytes. Now, looking at blood, plasma is one of 2 major components of blood. This is the liquid extracellular matrix of that tissue. And then you have the formed elements. These will include all the cells and cell fragments that are found suspended in the plasma. Okay, and if you were to actually take blood and spin it down in a centrifuge, what would happen is your heavier things, due to centrifugal force and gravity, are going to fall to the bottom. And that heavy thing is actually going to be erythrocytes. Now, the reason why the erythrocytes are heavier than your white blood cells is that the erythrocytes are going to have iron, okay? Because they have that iron present, what happens is that will actually make them heavier, and they will fall to the bottom. Now, as I just mentioned a few minutes ago, red blood cells are the predominant thing that we do find in blood, and they account for about 44% of blood. Leukocytes and platelets are actually going to be less than 1%, and that's what we are seeing here with this, that's what we're seeing right here with this buffy coat that you're seeing in this area right here, okay? Um, and then we have our plasma, which is going to be the remaining 50%. So blood contains 3 types of formed elements. They're going to be erythrocytes, which are your red blood cells, leukocytes, which are your white blood cells, and platelets. These are small cellular fragments. They are no longer functional cells or even resemble cells anymore. All 3 blood components or formed elements will actually derive from the same stem cell lines. There are 3 distinct layers that are formed when blood sample is centrifuged, as I mentioned. The top layer is going to be plasma. The middle layer, or the buffy coat, will be your leukocytes and platelets, and that is less than 1%. And then the bottom layer is the remaining 44%, which is your erythrocytes. Okay. Okay. Now, what are the functions of blood? Now, as you guys can imagine, some of your open-ended questions that you see is like, what are the functions of tissues or certain areas? So I would suspect that, you might see a question like this. I believe it's on your assignment sheet for the blood. But the blood functions are exchanging gases. Both oxygen and carbon dioxide are transported in the blood, not by the same mechanisms, but that is one of the major functions. Distribution of solids. Plasma transports ions, nutrients, hormones, waste products, and it plays an extremely important role in regulating ion concentrations in those tissues. Also performing immune functions. Both leukocytes and immune system proteins are transported throughout the blood, throughout the body within the blood as well. The additional following functions include maintaining body temperature, where blood can actually carry away heat that is generated by a byproduct of many of the chemical reactions that occur inside of cells. And also with the platelets and the various blood clotting proteins that are present in blood, you do have the ability to form blood clots as well. Now, the purpose of blood clot formation is that it seals damaged blood vessels to prevent any blood loss. Because, again, this is a closed system, and if there's any hemorrhaging or leaking within that system, that could be problematic. Overview of the blood functions. Continuing, we do have preserving the acid-base homeostasis. Blood pH is something that needs to be extremely well maintained, and it's a very finite range. Blood pH is maintained between 7.35 and 7.45. That's a fact you're going to want to know. This remains relatively constant as blood contains several important buffering systems. Any deviation, if you go below 7.35, you're entering into what we refer to as acidosis. If you go above 7.45, you're going into what we refer to as alkalosis. Both conditions can actually be fatal if they are not brought under control. Stabilizing blood pressure, blood volume is a major factor in determining blood pressure. I know some of the people that are more physics people might be able to understand this more than a non-physics people. But whenever you have volume, if you increase the volume, you're going to increase the pressure. If you decrease the volume, you'll decrease the pressure. The reason why blood pressure is so important, and we'll get into this, as we get a little deeper into the next 3 chapters here, is you need to have effective blood pressure in order to be able to perfuse the tissues. And if you don't have effective blood pressure, you may not perfuse all the tissues sufficiently, and then you can end up with death of those particular tissues. So let's look at the different components. Plasma, this is a pale yellow liquid whose volume is 90% water. This factor in determining viscosity of the thickness of blood falls on the plasma. The less water that you have, the greater the viscosity, and the less water that you have, the more sluggish your blood flow will actually be. Your plasma proteins, these will form a colloid, and they make up about 9% of the plasma volume. These will include things such as albumin, which is a large protein synthesized in the liver, and is responsible for the blood's colloid osmotic pressure. This is important because this is actually going to set up an osmotic pressure within the blood, as well as when you get to the level of the tissues, and how certain solids will move across the membranes. This colloid osmotic pressure will draw water into the blood by osmosis. This is because you are setting up that osmotic pressure. The difference is with those proteins being there, you're setting up an osmotic difference. You also have various immune proteins, gamma globulins, also known as antibodies, which are made by certain leukocytes and a major important component of the immune system. When we get to the immune system in a few chapters, we'll talk about antibodies, but they also do contribute to those plasma proteins. Transport proteins, these will bind to lipid-based molecules. We just got done talking about the endocrine system and how some of those hormones could be composed or derived from cholesterol or are lipid-based. Otherwise, if they did not get surrounded with these with these transport proteins, it would be incompatible to be transported in the blood, because the blood is mostly water, and that would repel those particular types of molecules. And then we have clotting proteins. These stop bleeding from injured blood vessels by forming blood clot with the assistance of platelets. All of your clotting proteins are actually formed by the liver. And that's why people that are in cirrhosis or have end-stage cirrhosis, they actually will have major clotting problems as well. Finally, the last 1% of plasma volume will consist of several small molecules, mostly dissolved in water portion of the plasma, forming a solution. These molecules can be readily exchanged between the blood and the interstitial fluid in most capillary beds. And this is a nice little table that just sort of summarizes all of the different components that we do find in plasma, okay? So just make sure you're familiar with what plasma actually does consist of. Cirrhosis, as I mentioned a few minutes ago, there are many causes that can actually lead to cirrhosis, including cancer, alcohol and drug abuse, especially over extreme periods of time, viruses. There are several hepatitis viruses that can lead to cirrhosis. And it might not even be alcohol and drug abuse, substance abuse issues, but if you have patients that are on an enormous amount of drugs in order to treat their various ailments, long-term exposure to those drugs can actually lead to cirrhosis. Unfortunately, that's what ended up happening with my own father. He was somebody who fell ill, in his late 50s and was on a lot of different types of medications. And as he approached the age of 70, for being on those medications for nearly 2 decades, he had pretty advanced cirrhosis at that point. So it's not just necessarily alcohol and drug abuse. There are many reasons that can lead to cirrhosis. Cirrhosis is common in the United States. It's actually the 10th leading cause of death for men, 12th for women. It results in a progressive decrease in the production of plasma proteins, which leads to the decrease of colloidal osmotic pressure, resulting in fluid loss to extracellular spaces, producing severe edema within the abdomen. We're called ascites, and typically these patients that have cirrhosis will start to build up fluids in the abdominal area. You also have a decline in the clotting factors, and this is a big issue because easy bruising, delay of clotting, this could be fatal if they're not able to actually clot anything as far as blood loss is concerned. And not just, hemorrhaging on the outside, but this could also be hemorrhaging internally as well. Okay, so next I'd like to talk about the erythrocytes, and then that's probably where I'll end part 1, as I'll continue, as we go through these lectures to break these up into different parts for you guys so that you can listen to small snippets there. So the erythrocytes, these are your red blood cells, and this is really an example of where structure and function really come to play very well. The shape and the components of the erythrocytes facilitate their transport of oxygen and carbon dioxide through the blood. Your typical erythrocyte or red blood cell is a biconcave disc that is flattened, donut-shaped cell, and it's concaved on both sides. And looking at the erythrocyte here, the reason why you have that concave structure on both sides is to increase the surface area to maximize oxygen carrying capacity. As the erythrocyte does mature, what happens is it's going to get rid of its nucleus and become anucleated. It will also lack most other organelles. Excuse me. This creates room in the cytosol for enzymes and nearly 1 billion oxygen-binding hemoglobin molecules per erythrocyte. And this, again, just really goes to the whole structure-function core principle that you may have been taught in biology. Essentially, in biology, we have structures that are designed to exist in a specific form so that, certain functions may accomplish, and that's really seen very well here with the red blood cells. Now, hemoglobin, this is a very large protein. It's actually a quaternary structure protein that consists of 4 polypeptide subunits, 2 alpha chains and 2 beta chains. Each polypeptide is bound to an iron-containing compound called a heme group, and an iron ion in each heme group is oxidized when it binds the oxygen in regions of high oxygen concentration, such as within the lungs, and that will actually form a molecule called oxyhemoglobin. So each hemoglobin molecule is going to have 4 heme groups and therefore 4 iron molecules, and therefore able to carry 4 oxygen molecules. So each hemoglobin has the capacity to carry 4 oxygen molecules. Okay. Hemoglobin will then release oxygen into regions, specifically tissues surrounding systemic capillary beds. Systemic capillary beds are going to be located all throughout the body. And this is typically where oxygen concentrations are low. This also binds to carbon dioxide, forming carb-aminohemoglobin, where oxygen levels are low. However, this is not how most carbon dioxide is transported in the blood. It actually only accounts for about 23% of carbon dioxide transport in the blood. Hemoglobin also has the ability to bind carbon monoxide to form carboxyhemoglobin. And this actually binds much more stronger and has a higher affinity than oxygen. That's why if your body is, posed with both oxygen and carbon monoxide is actually going to preferentially bind to hemoglobin. And when that happens, it changes the shape of hemoglobin, making it unable to unload the oxygen in the oxygen-deprived tissues. And essentially, when somebody has carbon monoxide poisoning, if it's not corrected pretty rapidly, that can lead to death. And it's often a silent killer, and it oftentimes happens, when people are sleeping. Now, the lifespan of a erythrocyte is relatively short. It ranges between 100 and 120 days. Due to damage incurred by the harsh environments in which they exist and any lack of means for repair, remember, they've gotten rid of their nucleus, they've gotten rid of most of their organelles, so they cannot undergo mitosis. What happens is the body must continuously make new erythrocytes. There must be a process in place for that, but you also need to deal with the erythrocytes that need to now be removed from circulation. Before we talk about how we remove erythrocytes from circulation, let's talk about how we form new erythrocytes. And that process is called erythropoiesis. Hematopoiesis is just a general term that takes place in the red bone marrow, where all formed elements in the blood are produced by the hematopoietic stem cells. Erythropoiesis is the specific hematopoietic process that produces erythrocytes from the hematopoietic stem cells. So again, all formed elements, which include erythrocytes, leukocytes, and platelets, are all going to come from those, HSCs, which are hematopoietic stem cells. Erythropoiesis takes approximately 5 to 7 days to complete. It begins when the hematopoietic stem cells differentiate into progenitor cells called erythrocyte colony-forming units. Once this happens, that is now a committed stem cell. What that means is that cell will now become a erythrocyte. The erythrocyte colony-forming units will then differentiate into proerythroblasts upon the secretion of erythropoietin. Erythropoietin is a hormone that must be present for erythrocytes to be developed. We did talk about this a little bit in the endocrine system, but the kidneys, which are not an endocrine organ, but they do secrete a hormone called EPO, which is erythropoietin, stimulates the production of red blood cells. Okay. So again, within the red bone marrow, the hematopoietic stem cell will differentiate into a erythrocyte colony-forming cell, which will then differentiate into a proerythroblast. The proerythroblast will then develop into erythroblast, rapidly synthesizing hemoglobin and proteins. The nucleus in the nucleus in the erythroblast will then shrink as it matures and eventually will be ejected out of the cell, okay, and as well as several other of the organelles, all right? And as this occurs, you're going to form what we call a reticulocyte cell. The reticulocyte cell then can enter into the bloodstream, where it will then continue to mature into a fully functional erythrocyte, which is a fully functional red blood cell at that point. Now, the regulation of erythropoiesis, erythropoietin or EPO, triggers negative feedback loop. So if the hematocrit decreases, you'll secrete EPO. When the hematocrit comes up, you will stop secreting EPO, okay? Again, hematocrit is the number of red blood cells that you have present. All right, and here's that little example there. The regulation of erythropoiesis, negative feedback mechanism. Now, erythrocyte death. Erythrocyte destruction takes place in the following steps. Erythrocytes become trapped in the sinusoids of the spleen, which is a organ in the upper left abdominal cavity. Spleen is essentially lymphoid tissue, which we haven't gotten to the lymphatic system yet, but it is lymphoid tissue. And it will contain splenic macrophages. And the job of splenic macrophages are to digest the erythrocytes. Hemoglobin will be broken down into, remember, hemoglobin's a protein, so it's going to be broken down into amino acids, which will get recycled, iron atoms, and heme. The iron atoms will actually be recycled, okay? Meaning that they will be used to make new hemoglobin in the red bone marrow. The heme, however, is not recycled for red blood cells. At first, it's going to be converted to a waste product called biliverdin, which is a greenish pigment. And then what will happen is that will be converted further to a yellowish waste product called bilirubin. Bilirubin is going to be very important for the production of bile, and we will talk about bile when we get to the digestive system, as that's where that really becomes important. Bile is going to be essential for the breakdown and emulsification of fats. Okay, and this just sort of summarizes the breakdown that we see with erythrocyte death. Now, some disorders that we see with erythrocytes, anemia. This is going to be a common condition. And essentially what anemia is you actually have a decrease in red blood cell or oxygen carrying capacity within the blood, okay? General symptoms of anemia include pallor, which is pale skin, fatigue, weakness, and shortness of breath. Many types of anemia cause elevated numbers of circulating reticulocytes. The body will then boost EPO production in response to diminished oxygen carrying capacity. Severe anemia can actually result in elevated heart rate. Body attempts to increase cardiac output to match the demand for oxygen from the oxygen-deprived tissues. Essentially, because you're not delivering enough oxygen to your tissues, those tissues are actually, sort of sounding off some alarms that, hey, we need more oxygen. And one of the ways that your body can compensate for that, especially, as, if you are replenishing those red blood cells, is to increase your heart rate, which would increase your cardiac output. Decreased hemoglobin, the most common cause for this is an iron deficiency. This is more common in females than males because as females go through their monthly cycle and they lose blood as a result, most people are not replenishing iron in their diet. So what can happen is that iron is lost if it's lost. It's not recycled back if it's lost. And if you do not have iron, you cannot make hemoglobin. If you don't have hemoglobin, you do not make functional red blood cells. Okay. There are also various types of anemia due to chronic diseases. Certain cancers can cause this, as well as, for example, vitamin B deficiency, malnutrition, poisoning with certain drugs, or heavy metals can also lead to this. Pregnancy can also cause different types of anemia. So the decreased hematocrit factors can reduce the number of erythrocytes in the blood to lower the hematocrit. One thing could be blood loss. Make sure you're not losing blood. Pernicious anemia, this results from vitamin B12 deficiency, which interferes with DNA synthesis of rapidly dividing cells, including those HSCs, the hematopoietic stem cells. Erythrocyte destruction can also be caused by bacterial infections, diseases of the immune system, or liver and lead poisoning, and this can actually result in hemolytic anemia. So here you're actually destroying more red blood cells than you're producing. Certain medications or exposure to ionizing radiation can also inhibit or stop the production of erythrocytes in red bone marrow. This condition is known as aplastic anemia and cause is oftentimes unknown. And then we have abnormal hemoglobin. This can cause what we refer to as sickle cell anemia. Sickle cell anemia is a genetic trait in which a single copy of a defective gene is asymptomatic, but if you have two defective genes, meaning you received it from both parents, you will end up with sickle cell disease. This is a normal red blood cell here. This is actually a sickle cell, and this is due to one base pair change in the DNA of the gene that leads to one amino acid change that completely affects the entire structure and function of this cell. When oxygen levels are low, red blood cells containing sickle cell hemoglobin change into that sickle shape. This leads to erythrocyte destruction in small blood vessels and reduction in the overall circulating erythrocytes. Okay, and this is where I'm going to end for part one. Okay, stay tuned for part two. Have a good one, guys.